Hello, fellow classmates and professors. My name is Angela Smith. I am doing Case Connections Unit 2 video presentation. And I am presenting case number 2, regulation of glucose uptake by the cell. And I am going to be talking about the uptake of glucose regulation by the following tissues: liver, skeletal muscle, brain, and adipose tissue, in conjunction with a glioblastoma tumor and fluorescence on a PET scan. So in the case connections problem number 2 is an MRI which shows a large glioblastoma tumor with areas of necrosis within the tumor. The tumor shows as a bright white fluoresced area within the brain field on MRI, and then after administration of fluorescently labeled D-glucose, there is enhancement of some of the boundaries of the tumor itself. So in normal tissue, glucose is upregulated, glucose is taken up in the following manner. In Unit 2, Week 4, Lesson 4.4, we learned about fuel metabolism and GLUT transporters specific to 5 tissue types. In this presentation, I will be addressing some of the GLUT specific transporters: insulin sensitivity and dependency, imaging, and specifically uptake of glucose comparative by normal tissue in the glioblastoma with necrotic tissue in the brain. So as I said, the MRI shows a large glioblastoma tumor, and I am going to be referring to the administration or the agent, the D-glucose, fluorescently labeled as FDG from here on out. And that's how it was referred to in all the articles that I referenced. So the phosphorylation of glucose to glucose 6-phosphate is catalyzed by either glucokinase or hexokinase, depending on the tissue that we're addressing. I learned that there are 4 hexokinase isoenzymes, 1, 2, 3, and 4, and 4 is also named glucokinase. In the liver, glucokinase or hexokinase 4 is responsible for catalyzing the phosphorylation of glucose to glucose 6-phosphate. The GLUT2 transporter will uptake glucose in an insulin-independent manner. Skeletal muscle, hexokinase 2 is the catalyst for phosphorylating glucose to glucose 6-phosphate, and the GLUT4 transporter will uptake glucose in an insulin-dependent manner, meaning only if there's insulin present, such as in the fed state. In the brain, hexokinase 1 phosphorylates glucose to glucose 6-phosphate via glycolysis using GLUT1 and 3 transporters in an insulin-independent manner, and it will always preferentially oxidize glucose except in cases of extreme starvation. In the adipose tissue, hexokinase 2 phosphorylates glucose to glucose 6-phosphate and will uptake glucose in the fed state via GLUT4 transporter as well. This is also in an insulin-sensitive or dependent manner, the same as the skeletal muscle in terms of the transporter and sensitivity. So the question was posed: how does glucose uptake in normal tissue compared to glucose uptake by tumor cells in the presented image, and why would this be advantageous to the invasive tumor? And what I found was that normal tissues have a slower uptake of glucose than that of malignant cells. Malignant cells have increased rates of glycolysis and larger number of transporters on the cell surfaces, transporter proteins on the cell surfaces. The consumption of glucose by the malignant cells works out well for them because of the brain's preference or affinity for glucose. The faster rate of glycolysis, as well as the increased glucose transporter proteins on the cell surface, facilitates the detection of cancer cells using the FDG in conjunction with PET scanning technologies. And I guess this has been used for a while. Now, I did find a more primitive type of FDG, which I think was 2-diglycol phosphate or some former relative of the FDG that is currently being used. The metabolically active tumors appear hot on the scanned images, meaning they fluoresce a brighter white color, radiopaque, if you will. FDG is phosphorylated to FDG6 phosphate by hexokinase after it enters the cell. It cannot be metabolized further in the pathway of glycolysis, and it stays trapped in the cell. Malignant cells do not contain enough G6 phosphatase, glucose 6 phosphatase, to be able to reverse the phosphorylation. Hence, they get trapped in the cell. What modifications, the next question was, what modifications may have occurred in cancer cells to alter glucose uptake as predicted above, or in the presented scenario? What I discovered is fluorescent D-glucose is a radiotracer molecule that acts or behaves like glucose and is commonly used in PET scans for detection of malignant cell proliferation. In other words, multiplication, or the proliferation of the cells. The structure differs from glucose so that it is not systemically absorbed or further metabolized. I found an article that suggested glutamine metabolism is upregulated on the oncogenes or cancer genes as well. And many cancer treatments target the glycolytic pathways of the malignant cells. It's also suggested that treatment be aimed at both glutamine and glucose pathway integration. So in other words, targeting both pathways, interrupting both pathways, or tagging the treatments along with the uptake of the glucose and the glutamine in the cells is most effective at treating glioblastomas. Thank you. And then the next case I presented or I addressed was the case number 3, a 5-year-old boy who presented as described in the scenario, is likely in diabetic ketoacidosis, secondary to new onset type 1 diabetes. Diabetic ketoacidosis is a life-threatening condition where the insulin ceases to be produced by the beta cells in the islets of Langerhans in the pancreas. As the insulin is unavailable to facilitate glycolysis, the serum glucose levels rise and ketosis begins. The electron transport chain can be affected and lactic acidosis can result as well. The patient would likely be tachypnic, in an attempt to blow off CO2 and autocorrect or self-correct for acidosis, and would likely have fruity breath, another hallmark of diabetic ketoacidosis, secondary to the ketones produced being expired in the respiratory cycle. He would likely be tachycardic and have ketone bodies in both urine and blood. The lack of insulin results in increased protein and triglyceride metabolic release from skeletal muscle, and the increased triglycerides release glycerol, and the increased protein metabolism releases alanine into circulation. Both of these substrates are used for gluconeogenesis in the liver, which is upregulated in the absence of insulin, and also with the elevated glucagon. The enhanced rate of gluconeogenesis coupled with the inhibition of glucagon-mediated glucose storage into glycogen results in an increased release of glucose from the liver, and hyperglycemia is a resulting condition and can have systemic consequences such as electrolyte imbalances at kind of the mild end of the spectrum, cerebral edema caused by an osmotic pull at the other end of the spectrum. And then there is also a condition known as HHS, hyperosmolar hyperglycemic syndrome, or HHNK. There's a couple different names for it. HHNK is hyperosmolar hyperglycemic non-ketotic condition. Is also a complication of diabetes, but is typically seen in, I believe almost exclusively seen in type 2 diabetic in adults, and is associated with high mortality, extremely high blood glucose levels, sometimes off the scale, at least as far as like glucometers go. It may just say high, critical high, and sometimes in the thousands range. There are no ketones produced in these patients. Management of a type 1 diabetic DKA pediatric patient is somewhat dependent on the presentation, and I did draw the majority of this information from my own prior experience working in the PICU. It is best to avoid intubation, as it's often quite difficult to match what the patient's body's doing on their own in terms of their minute ventilation. Their respiratory rate is going to be elevated. The body's attempting to breathe off the CO2 in hopes of autocorrecting acidosis. An insulin infusion is most certainly a given, but is pretty cautiously titrated. You don't want to drop their blood glucose levels too rapidly, causing fluid shifts and further issues. And then we used a recipe, if you will, where we took a liter bag of D10 with 20 milliequivalents of potassium chloride, or sometimes potassium phosphate, KPhos or KCl, and then a liter bag of normal saline without any sugar in it, and 20 of KCl or KPhos, and wide them in together to be able to titrate specifically the amount of dextrose that the patient would receive. Again, you don't want to drop them too quickly. You could have resultant hypokalemia, hyperchloremia. The electrolytes going in and out of the cells can create a problem. So correcting can be a bit of a challenge. And then the insulin is typically on a separate pump. Metabolic panels are drawn initially every 2 hours with blood glucose finger sticks in between. So every hour you get a blood sugar level on these patients, and it can be a bit of a challenge, but I actually enjoyed the management of the DKA patients and the teaching of the patients and the families on how to manage Nuancet DKA. So I do have my references that I will submit via Dropbox, and thank you very much. I appreciate you taking the time to listen to my recording. Thank you.